For introduction, the understanding of the relationship between organism and the environment is fundamental to elucidating the dynamic of the ecosystem. The distribution and abundance of organisms are influenced by a lot of factors ranging from abiotic components like temperature and precipitating to biotic interaction such as competition and predation. Biotic factors in the ecosystem encompass all living organisms present within the environment. When organisms of the same species inhabit a shared environment, they form a population. Objective of the experiment is first to introduce the simple concept of quadrat, second to enhance the power of observation, third to record the difference in plant distribution within the quadrat, and lastly to infer from natural observation of the possible factors that affect distribution and abundance of organisms.
The material needed for this experiment are raffia string, notebook, scissor, stick, and the last one is the measurement tape. For this practical tool, UiTM Pahang Campus Jengkau Forest will be used as the area under study.
The quadrat of 5 meter times 5 meter of each forest type were made. Multiple quadrat samples will be performed throughout the habitat at several random locations.
All the tree stands that are found inside your quadrat were observed and take note. The plant species in your quadrat area were selected and being collected. All the plant species data collected in your quadrat were tabulated in the table. The data will then be used to estimate the population size and population density within the entire habitat.
Based on the result shown, this is the result that we were taken from practical 2.
What is the difference in the number of plant species in the quadrat? Hypothesis or make your own assumption. Discuss the factor that would have caused this type of distribution. There may be several factors that impact on the variation in the number of plant species in the quadrat, resulting in uneven distribution. To explain this kind of distribution, a number of theories of presumption have been made. First of all, environmental heterogeneity in which change in soil properties, moisture content, solar exposure, or microhabitat within the quadrat may allow the emergence of distinct plant species. Certain conditions may favor some species more than others, resulting in their dominant or absent in particular location. The factor might include competitive exchange. Plant species distribution may be impacted by competition for resources like nutrients, water, or light. Lower species richness in some section of the quadrat may be caused by dominant or aggressive species that outcompete or hinder the growth of other species. Some plant species may have limited dispersal capability, which might affect their distribution within the quadrat. Variation in species composition can be influenced by factors such as seed availability, dispersal method, and dispersal obstacle. The next issue to consider is biotic interaction, in which interaction with other organisms such as herbivory or mutualism can be impact the distribution of plant species within the quadrat. Herbivore, for example, may preferentially devour certain plant species, resulting in their depletion. Finally, there may be a random chance component in the distribution of plant species inside the quadrat. The interaction of environment element and ecological process can be complicated, resulting in a stochastic pattern of species distribution.
For the post-lab question, define the term quadrat in practice. Quadrat is an ecological sampling unit consisting of a small square frame that is laid down in which all species of interest or specific area of the community to be sampled and the measurements are taken. Within the quadrat frame, the existence of plants are recorded using an appropriate measure of abundance. For example, it can be used to measure the abundance of various plants within the quadrat frame. Second, What are the two basic requirements of all techniques? First, area of the quadrant must be known. It is essential to know the exact area of the quadrant being used in the sampling technique. The area of the quadrant is typically determined by measuring its dimensions, such as length and width. Knowing the precise area allows for accurate calculations of population density or other ecological measurements within the quadrant. Second, the population in quadrant must be determined exactly. This is because to calculate population density using a quadrant, the number of organisms or target samples within the quadrant must be determined precisely. This requires careful counting or identification of all samples belonging to the target species or organisms within the target. Third, what would be the advantages and disadvantages of using a larger or smaller quadrant to survey the sedentary organism? Disadvantages is used to survey the sedentary organism that is inexpensive because it only requires few materials. Besides that, it is a feasible method and easy to use compared to other sampling methods since quadrant plots are uniform in size and shape and distributed randomly throughout the sample area, which makes the study design straightforward. Apart from that, larger quadrant can give more accurate estimation of overall densities, but it can be complicated to carry out due to the time consuming to place and sampling them. For these advantages, in spite of the relative ease of designing the quadrant, there might be study errors. Quadrants that are too large, too small, or spaced inappropriately often result in errors. For example, randomly spaced quadrants which are too small might miss too many individuals to be counted, which will result underestimation of actual population size. Other than that, inconsistent counting and omitting species that lie partially in boundaries also can occur. For the last question, which is number 4, problems with spatial scale often make it difficult to directly estimate average densities in habitats such as large forested areas or in aquatic habitats where one cannot directly see the animals or plants of interest. Explain the issues concerned ecologists in conducting the sampling method. Problems with spatial scale make it difficult to estimate average densities in large forested areas or aquatic habitats due to inaccessibility, which can be challenging to access, making it difficult to perform on-site sampling. The second factor is heterogeneity, which is frequently strong in this ecosystem and characterized by variance in the vegetation, microhabitats, or water equality. The distribution and abundance of organisms may be affected by this heterogeneity, which make it difficult to collect representative samples. Next is a scale mismatch. The scale of the sampling procedure may not match the scale at which the organisms are spread or interact. For instance, if an organism displays fine scale patterns or interactions but the sampling method uses a wider scale, the samples may not be accurately captured. To conclude, quadrant sampling does help in understanding and determining factors that affect the distribution and abundance of organisms. By systematically sampling across a different microhabitats, we gain a comprehensive understanding of the community composition and structure. From this, practically, distribution and abundance of organisms are influenced by a lot of factors like the abiotic conditions and the biotic interactions. Abiotic factors, including temperature, moisture levels, and light availability, were found to significantly shape the suitability of habitats for various organisms. Biotic interactions such as competition, predation, and symbiosis, on the other hand, played a pivotal role in determining organismal distribution and abundance. Overall, this practical emphasized the complex interplay of abiotic and biotic factors in shaping the distribution and abundance of organisms within ecosystem.